Okay, I just started recording this. So hello everybody. Hope you're here for the vertebrate zoology class. Let's see. I recognize most of you, but not everyone. I know we have some students on the East Coast, and we have some military students that are not going to be in class. They're going to be listening to the recording. So hi to everybody. So my name is Mario Mota. I'm your professor for this class. Let me start sharing the screen for a second. Can you see the Blackboard page for the class?
Yes.
Yeah. Okay. Thank you. So this is the class in Blackboard. I'm guessing all of you know how to get on Blackboard. I know some of you are not are new to National University, but in the syllabus that I emailed you, there's information on how to get to the Blackboard, and if you have problems, who to contact, the student concierge. One of the things I recommend, and now again, if you're used to National, this is old news, but use Chrome instead of another browser. It's just better and doesn't crash like the other ones do. Okay. Now, for other classes, you might have used Collaborate. Some of you were in Dr. Maxwell's invertebrate class, right? I know Colette was there. Yeah. Did you guys use did you use Zoom or Collaborate?
I will use Zoom.
Zoom, okay. Yeah, Zoom is better because Collaborate takes, like, I have a lot of videos to show you, and it takes forever to load them into Blackboard. So what I'll do is we're meeting in Zoom, and I usually stop recording after an hour and then start again. So I have 1 hour of videos, and I'll post the link in Blackboard. Okay, so the actual video will be on YouTube, and I'll give you the link. That way you're not going to slow down or have all these problems with the Blackboard. Okay. So when you're looking at this page, look, can you see the mouse?
Yes.
Yeah, okay, thanks. On the left side, you see that this has 8 weeks. Normally, this class is in January, and then the lab is in February. So we combine them into 8 weeks instead of 4. And the way we teach this, or the way I teach this, is there's every Tuesday and Thursday, there's a lecture for about 3 hours, and then we do the lab. So every day we have a lecture and lab. Because of all this COVID mess, we can't do that. So we separated the lecture for now, and then we're going to do the lab in February. And as of now, it's still going to be in person at the Spectrum. At least that's what we're planning. It could change. It's probably going to change. No one knows. I'll be honest, it's a lot more fun in person because you get to dissect animals, and we go on field trips. We go to the zoo, the aquarium, we go bird watching. But if we can't, we can't. I know that there are some students that need the lab these classes to graduate. I mean, some of you do that. So what I'm going to do in January as a backup is I'm going to film the dissections and things that we would be doing in the lab, just in case. If we can't have the class in person, at least there will be something. I can tell you that I was in the Spectrum about 2 weeks ago, and they are setting up the plastics, not the shields. So that big, if you're familiar with the biology labs, the the one that we call the micro lab has all these plastic shields, so everybody is separated from other students. We're only, I think the maximum students are, I think 12 or 15, because they're all spread out. So there's no one's next to each other, and then there's 6 feet separation next to you and behind you, so that they are setting up for that. Okay. Okay, Joe just said, yeah. I just told you that they are making the putting those separations there. Um, I don't know about if they're going to force you to do that or not. They can't force you to take the class, but right now, to do a dissection with your hands, having the animal in front of you is very different than doing it or watching a video. So it's we'll see what's going to happen. Some of the other trips, like going to the zoo, you can go on your own. You don't have to go as a class. Go to the aquarium, and I might also go and film so that we'll have videos for you to see. Okay. But between now, I'll be honest, between now and February, everything is going to change, especially since we might have a vaccine ready at the end of this month. We'll see. Okay, so what I was telling you about this, when you see this on the left side here, the 8 weeks, don't don't get worried. We are not, we're going to have 4 weeks, and except that this is the Blackboard shell, the master shell that we use for this class. So if you look into those weeks, if you click on them, they're empty, and I'll be moving things in and out of from the 8 weeks to the 4 weeks. Okay. Now I know if you're looking at through your computer, you're not seeing the same thing I have. Some of the things I have hidden because I'll open them up when it's time to show them. Okay, but you should see. Actually, this is what you see. Hold on. You should see these folders. Okay, and these are the PowerPoints that I'm going to use to lecture. Okay, so you can download them, follow along, or just leave them here. So you see like 1, 2, 3, 4. Okay, this is actually a handout for you. And then on the last one, this is a folder that's got links to 3 YouTube videos. This is one of the things that I would be doing in the lab. We will be watching this, okay? But it's actually part of the lab, of the lecture. And this is going over the kind of like how life started. How did planet Earth get animals and plants? What happened? And we're not going to watch these in class. I'm going to leave them here, and watch them when you have time. They are 10 minutes each, so you have 3. Okay, and then I have a study guide here. Okay, because this is one of the quizzes will be on this. So if you use the study guide to help you take notes when you watch the videos. Okay. Okay, so that's why we have these 8 weeks. Okay. Now, before I go through the PowerPoints, I want to go over the syllabus just to make sure that if you have questions, I'll go over them. So let me stop sharing and share the syllabus. Okay. Now you should see this, right? It's the, okay, so this is the same thing I emailed you. It just has the general information on the class. Um, here's the information for Blackboard. Okay, and I don't know if you had a chance to read this. If you email the faculty or the student concierge, they send you. There's an automatic email that you get back saying that we received your email, all that. Then get a ticket number, because sometimes they have to escalate it to the next level, and without that ticket number, it's really you kind of get lost in there. Okay. Now, if you want to reach me, use the email. It's really the best way. And if I don't email you back right away, just resend it. I usually check it in the morning. Usually our mornings are meetings, and then we have the afternoon to teach. So in the mornings, I'm usually, I can't do anything if I'm in the meeting. But then in the afternoon is when I go back and read the emails. Okay. The textbook. Some of you told me you already have it. I know, or if you're new to National, this is National is a 1 month, our classes are 1 month long. So it's really intense. And we don't expect you to read everything in the book. You don't have time for that, okay? Study what I go over in the lectures. And then if you don't understand something or you want to read more about a particular thing, go to the book, okay? But don't try to read everything in the chapter, because we might not cover everything in the chapter. And then you maximize your time. Study the notes from lecture first, then go to the book. Okay. There's learning outcomes. So as far as grading, there's 2 exams and a final, and then 2 quizzes. Okay, and I'll show you how we do them in a little bit. Okay, you're familiar with the grading assignments. Obviously, turn things on time. Okay, so here's the schedule, and again, this might change depending on what happens each week. Okay, so when you look at the dates on the left side, this is the week, not the day. So the week of November 24th, week of December 1st. Okay, and again, we might have 2 classes or 1 class like this week, or some weeks we'll have 3 classes. We'll have the Saturday. And the reason for Saturdays is because of Christmas, we're going to end the class earlier than going until the 31st. Okay, so this week we only have class today, no class on Thursday. Okay, and this is what I have planned to teach tonight. Okay, this part right here, this is what the videos are going to help you with this. Okay. Then next week with class Tuesdays, Thursdays, and Saturday. Okay. Now on Tuesday, I don't think I can be here in person to teach you because I have an MRI at 4:30, and I don't know. It's the ones where they have to inject you with fluids. It's my knee. So I don't know if I can make it on time. So what I'm going to do is I'm going to record the lecture on Monday and post it. So if I can't make it here, I'll just basically I'll have the lecture there that will be hidden. If I can't make it, I'll open up and you guys can hear it. Okay. And then you don't have to listen to it on Tuesday. You can listen to it whenever. Okay. But I'll try to make it. And all these funky words you see here, strachoderms, placoderms, gnathus, stomata, all of this is fish. So we're going to start talking about vertebrates this week, learning how life appeared on the planet, and then start with fish, and then just move our way up to amphibians, reptiles, birds, and mammals. Okay. The quiz 1 and quiz 2 We will schedule it for next week. Again, quiz 1 will be based on those videos and that study guide. Okay. And I'll give you time to hear them, listen to them, and study. Probably will be on Wednesday. And the way it works is I'll post it on week 2, and I'll give you all day. So you know, like say the quiz might be like 20 minutes or half hour, but you can take it anytime during that day. Okay. But once you start it, you only have half an hour, but you don't have to go and take it at a particular time. That way, you know, you can take it during your lunch, at night, whenever it's, and people in different time zones, you can take it when it's not midnight. You know. The exam's the same thing. So we have an exam basically on every Saturday. Okay, so exam 1, exam 2, and then final. So the exam will be opened the whole weekend, and you take it whenever you want. So it will open Saturday morning, and you have until Sunday, 11:59 p.m. Pacific time. Oh yeah, if those of you on the East Coast, all the times I post are Pacific times, so just keep check of that. Okay. So the exams are open again from Saturday morning to Sunday night, and depending on how much time you have for the exam, again, once you started, you have to you can't pause it. You have to take the exam, but you can do it any time during the weekend. One thing though, let's say if you only have 1 hour to do the exam and you start at 11:30, that means you have to be done by midnight. So you don't get an extra hour, an extra half hour. Okay, so keep in mind, you have to be done by 11:59. Actually, I think Blackboard gives you like 15 minutes grace period and a lot of warnings, but just make sure you kind of plan the time, you know, to get done. Okay. And then that's all I think. So this, and I'll give you more updates on this. So the exams will be on the weekends. You take them whenever it's convenient for you. And then the quizzes will be next week. And again, I'll let you know what day, and you take it whenever time you want it that day. Okay. So any questions on that? No? Okay. So then all of this at the end of the syllabus is standard for National University. But if you're not a student of National, or if you're new, this is something that, you know, read this and email me if you have questions. Again, this is just going over the exams, what I just told you. Let me see what else. Yeah, all of this is standard. Okay. All right. So if no questions on this, let me stop sharing, close it. All right. So if you go to the Blackboard, okay, I'm going to start with the file that says the vertebrate zoology. The first one says what makes a vertebrate. So that's the one I'm going to start. If you want to follow, download it or not. I'm going to share my screen. So mine is a little bit different than yours. I just have more slides. And a lot of the slides that I have that you don't are just basically pictures. That's why I didn't want to give you a whole bunch. So let me share this. Again, if you have questions, just, you know, either type them in or just let me know. Now, I know most of you, a big number of you, just finished the invertebrate class. So what I'm going to go over in this PowerPoint right now, this is something that you had already, that you just had it, you just finished. So this is for you will be a review. It's kind of like a continuation, or of the last class. We overlap the classes, but not everyone here in this class had the invertebrate class. So for those of you that this is new, if you don't understand something, just let me know, because I tend to go a little faster on this part just because it's a review. Okay. So you can see the PowerPoint with the snake skeleton. It says vertebrates, correct?
Yes.
Yeah. Okay. Okay. All right. So again, so this class is about vertebrates, and those of you that were on the last one, that was invertebrates. Okay. This is the nicer class. These are the cooler animals. They have a backbone. You can relate to them because they're like us. They're not like slimy little things like earthworms and all those worms. But when we start studying on the vertebrates, we're basically building up from the invertebrates. So we're taking what had evolved, the invertebrates, and they are just kind of think of like improving them, or taking what they did, what they had as far as the anatomy and their physiology, and they're evolving them and adapting them to other environments, especially um, when they get big and walk on land. Okay. Um, when we talk about vertebrates or life, we have to go back to a period called the Cambrian period. Actually, let me look at the in Blackboard. There is a handout called um, geological time. Uh, that's the one I'm going to share right now. Okay, it looks like this. Okay. Now you might have seen this again in invertebrate class or other classes. This would be the history of the world. Just imagine it's this. Okay. The oldest is on the bottom here, and then the most recent is on top. Okay. So if you go across the top, you got eons, eras, periods, epochs, and then MY is million years. Okay. And if you look at the scale, you see that it's not like years broken in smaller portions. And then when you get here, it's like 500, 2,000 years. Okay. So basically this is the history of life and not life on the planet. And if you could take the 4,600 million years, and which are here, this part right here, the Precambrian, this would be 7/8, and then this whole part would be 1/8. Okay. And we like humans, we show up right here on the end of the Holocene. Okay. So it's most of the history of the planet, which was here, there wasn't really any life. Okay? And that's what those three videos are basically going to show you. They're going to go over this what happened. Okay. How the planet formed, how life came to evolve. Okay. But the very, and by the way, it's a really cool videos. The animations are really, really, really nice. But basically, the life appeared when, as far as we know from fossils, in the Cambrian. Okay, and it went from like having nothing, the planet having no life, to bang, there's a big, all these animals, and these were in the sea. So life began in the ocean. Okay, So we call this the Cambrian explosion because it looked like an explosion, like there was nothing, and then boom, there's all these animals. Okay, and so Cambrian is the oldest with life, and then as we move up, we go to Ordovician, Silurian, Devonian, and then this one is the Carboniferous, and it's divided into two, Pennsylvania and Mississippi. And by the way, these are only used in the United States, obviously because of the two regions, Pennsylvania and Mississippi, and then Permian. Okay, this makes up the Paleozoic. Okay. This whole thing, the Paleozoic, is referred to as the age of fishes. So in the Paleozoic, fishes. That's the ruling animal class. Okay. Then we go to the Mesozoic. It only has three: Triassic, Jurassic, and Cretaceous. This is the age of reptiles. And you remember the movie Jurassic Park, the dinosaurs. Okay, so Mesozoic was reptiles. And then we go to the most recent one, the Cenozoic. And here we have mammals. So mammals are the ruling animals now. Okay. And by the way, birds are considered reptiles right now. Okay. So they're basically, right now, they are the living descendants of dinosaurs. Okay. So don't worry about these names because we will go through them and explain what they are. But I wanted to show you this before we start, because a lot of time of this, of the time of the planet, our planet, there was no life. That's the pre-Cambrian. Okay, and again, the videos will show you that. It was only about when the Cambrian came that we have fossils, we have life. Okay. But just a side note, what separates these, the Paleozoic from the Mesozoic, from the Cenozoic, are extinctions. So big extinctions is what makes these separations between these two eras. Okay. All right, so let me go back to the other PowerPoint.
Okay, sweet, perfect moment. Okay, babe, headphone. I'm gonna toss it at you. Toss it over you.
Can you mute? Brianna, can you mute? I guess not. All right, so here's the vertebrates. Okay, so again, just think of the vertebrates evolving on the invertebrates, on the body plan and the anatomy and biology of the invertebrates. Okay. I don't know if all of you had genetics or not. I mean, let me ask, how many of you had genetics?
I have.
Yeah. Yeah. Okay. So, so two, but there are these genes. I'm going to type them in here, called Hox genes. Okay, and they are basically, in a simplified version, they are like a supervisor gene. So the Hox genes tell other genes when they get turned on and do whatever they do, when they get turned off. Okay, and the number. So basically, these Hox genes, and this is something that was found within the last 10 years. This is something new in biology. But these Hox genes are very similar, or almost identical in us, plants, across the animal, the different species. So it's something that's been conserved throughout evolution. Okay. And when they looked at the Hox genes of vertebrates versus invertebrates, we see that it's the same genes that have duplicated in some cases. So imagine like an invertebrate has 4 Hox genes, and now you have 8. So you have all these extra genes that can evolve in different ways. So that starts to explain why the vertebrates are so diverse. There's so many different vertebrates, from different sizes, from like the big whales to a little shrew, but also from underwater, the air, land, underground, is just very, very diverse. Okay, so the idea was that the basic traits were built on the invertebrates, which means they were built on the Hox genes that invertebrates had. Okay. And then these are, so you imagine you take all the invertebrates, all the vertebrates, sorry, and you try to think of, okay, what would be some things that they all have in common? How can we come up with a criteria of what makes a vertebrate? Okay, and as of now, we have these 5 things. Again, if you had invertebrate biology, this is new, but not all of you had. I can tell you one thing, when I was going through school, there were only 3. Now there are 5. So they're always finding new things, okay? So these 5 things is what makes or classifies a vertebrate. So I'm just gonna go quickly through them, okay? So the notochord, again, all vertebrates have this at some point of their life. Some might not have it as adults. They might lose it, but at some stage, it's there. Okay, so and that's the important part. At some stage, or doing embryology, it's there. Okay, so notochord, and on the dorsal side, the notochord is still present on a group of animals called the hagfish. I'm going to type it in here. Hagfish. Okay, it's a fish, obviously. Looks like a lamprey. Okay, and we will study them. Okay, what the notochord develops into in other vertebrates is basically the discs between the vertebrae, the vertebral column. Like you hear someone has a slip disc or a crushed discs, that's the old notochord. Okay. Number 2, the dorsal hollow nerve cord, basically the spinal cord. Okay, and that is the spinal cord. Okay, a tail. Now it says post-anal tail because it's post the anus. Now the tail, some animals have it. You think of dogs and cats. Others don't, like us. But if you look at at our skeleton, we still have the bones there, the coccyx. So we still have the bones of where the tail was. Okay. Pharyngeal gill slits. It's a big fancy name, and it's just during the development, we have these gill slits. Think of like a fish. That's what these little holes are. Gill slits on our throat. Okay. Now, in invertebrates, you see this a lot. That the invertebrates that have gill slits will have little openings for filter feeding. The water comes in, and the water carrying food, and then they have something. Hold on, I got drinks. Hold on a second. 1 second. Throat was going dry. So they have something that will filter that food, the water, and catch the food. In vertebrates, the only animals left with gill slits are the fish. Okay. In us, or mammals, birds, reptiles, all that, this has become part of the hearing, part of our jaw support. So it has changed to something else, like part of the throat. The endostyle, this is a little gland. Think of it our thyroid. Okay, so this would be the homologous or the version of the thyroid in other animals. Okay, so here they are. Okay, so again, this is just to give you an idea that these are the 5 characteristics that makes a vertebrate. Okay, now the again, what we learn in this class, or what every year I have to adapt this class and not adapt, upgrade this class because there's always something new. I know a new monkey just discovered 2 weeks ago. There's always coming up. But a lot of what we know from the old biologists and taxonomists and zoologists was based on what we call the morphology. Basically they looked at fossils, they looked at animals, and they try to make some sense. And then came this new thing called genetics, and that damn Mendel found figured this out, and then they started looking at genes, and then they went back and started reanalyzing what these old biologists did based on just looks, morphology, and they saw that the genes were telling a different story than what they had. So the world of taxonomy and what we thought about the way the relationships of the animals got, and it's still being changed upside down. For example, the mammals, I'm jumping way ahead. Mammals had one big group called insectivores. So it was basically like all the little animals that eat insects. They were all together. Okay. And then the genetics were like, oh, no, they are completely different. So now the insectivores doesn't really make sense. Animals were taken out and put in different groups that don't, and these animals don't look alike, but the genes say that they are related, and again, the old biologists wouldn't have known that. But for a while, it was kind of like sad to see that all the work that these biologists had done was being thrown out because these new young biologists were coming in with genetic tools and saying, "Oh, you're wrong," So it's still ongoing. All right. So where I'm going with this is the vertebrate tree. Okay. So if you had invertebrates, you finished with the echinoderms, the sea stars, and then you started the vertebrates with these animals that are kind of like vertebrates, but they're like primitive vertebrates. Okay, and one of the groups, they are vertebrates, but not what you consider the true vertebrates. Okay, so again, chordata are the vertebrates. So the two groups, the urochordates and the cephalochordates. Okay, again, big names. The urochordates, the common name are the tunicates. I'll show you what these look like. Okay, and then the okay, I thought that was misspelled. No, cephalochordates. The Amphioxus is a genus of a really common one. The common name is lancelets, and you probably studied them in invertebrate. Okay, these are the tunicates, sea squirts. They look like sponges. When you look at them, they look just like they're in the water. They don't they're marine. They look just like sponges. I work with sea turtles, and some sea turtles eat these. And when you see them eating them, you don't think, oh, that's a tunicate. It just looks like something green full of algae. They're not as pretty as these, okay? But in the larval stage, okay, so here on the right side is the larval stage. You see that they have the those characteristics that makes it a vertebrate. Okay. The other one, the cephalochordate, again, the Amphioxus, it's a little, I mean, this looks long, but this animal is probably 1 inch, really small, and it buries itself in the sand. The head, slide this, the anterior portion is sticking out, okay? And again, the animal basically just feeds off the water, okay? There's not much to it, but when you look at its anatomy, okay, here's a simplified diagram. You see that it has those characteristics, okay? And here on the left side, you see how small it is, okay? So again, this is just a fast version to let you know that we had these, we are in the chordates. We have, let me go back, okay, the urochordates and the cephalochordates, okay? So these are the transitions basically from the invertebrates to the vertebrates, okay? And then we get to the chordates, the true chordates, the true vertebrates, and then we start with fishes, okay? So I'm gonna, any questions before I close this and go to the next PowerPoint? By the way, I know I went a little fast on this because this is a little bit of a review, okay? Okay. So let me bring up. So now I'm going to jump a little bit. I'm going to go how fossils are formed. So if you want to open that PowerPoint, and I will share it. And by the way, we're not going to be here until 10 o'clock. I usually teach for like an hour, hour and a half, 2 hours, because I know that, especially if you're on the East Coast, you're going to be going towards midnight. Okay. So before we go into the actual, you can see this, right? You can see the PowerPoint with fossils?
Yes, we can.
Okay, thanks. If I keep asking because sometimes you might not see it and I just keep going. Okay. So before I start talking about fishes and all of this, I just want to go back a little bit and give you some background on fossils, how they are formed and the different ways they're formed, because basically what we know of that time that I was telling you, the Cambrian and all that time, is from fossils. So because of the way they form, it's very biased. So that there could have been other animals, but we just don't know because there are no fossils. There's no record. Okay. Oops. Okay. So, and again, you have these on Blackboard, on this PowerPoint. Okay. So basically, fossil is just the imprint of an organism that lived a long time ago. Okay. So the animal died, and the body became imprinted like this on the rock, or a rock, the water with minerals infiltrated the body and formed a cast of the animal or plant. Okay. So first thing is, I'm going to go through ways that fossils are made. Okay. The most common one are these fossils in the rocks. Okay. And this is just easy when an animal dies and usually it has to be near water. Okay, so a lot of things have to happen to have a fossil. First, it has to die. It can't be eaten by a predator. It can't decompose. Like if it dies on an area that's really hot, it will just decompose, and be destroyed. So it has to die near water so that its soft body parts are not eaten or destroyed. So near water tends to preserve this. And also not just near water, but near a bottom, like a soft sediment, like a sandy bottom that will kind of hide. The animal will sink and it will get covered and preserved. Okay. So just think about we know we have a lot of fossils from water, from oceans, from rivers, just because it was easier to fossilize than on land. So it's, again, kind of biased. Okay. So what the process is, just imagine you have this fish, died, went to the bottom, okay, and sinks to the floor. The soft parts of the animal didn't rot away. The animal is buried by the mud or sand, so it wasn't eaten. And again, I think also like anaerobic decomposers like bacteria that didn't destroy them. Okay, so then the skeleton is buried, or the animal's buried, and then the sediment is added on top. So if you look on this one, the animal's covered by sediment. Okay. So again, with the pressure of the sand or whatever the sediment is, the sediment turns into rock. And this is what this big word here, lithification. Okay. So when the sediment turns into a hard rock. Okay. So now the skeleton or the whatever it is, the animal will just dissolve and then leaves a mold, a cavity. And then the water has minerals, and then the minerals will get deposited in that cavity, and then they leave the shape of the animal. Okay, so you get the mold. You don't know what was inside in this type of fossil. You don't have the oops, sorry. Oh, sorry. There you go. You don't have the internal organs, okay, but you just have the kind of like a cast of what was there. Okay. Again, the other word, this other word, permineralization. Okay, it's something similar where the minerals in the water, in the ocean water, will get deposited or get caught in the body of this animal, and then form what was there, but in rock, okay, in minerals. And again, you need a lot of luck that the animal has to die, sink, not get eaten, sink to the bottom. The bottom has to be a soft sediment. The conditions have to be right so that it doesn't decompose and gets covered and then gets turned into a fossil. Okay. Another way is amber. Okay, and this is usually, well, not usually, it has to be with something small like an insect, a frog, lizards, and they're just caught in tree sap. And then the sap hardens and a fossil of the animal is created. Okay, so this is called amber, but it's basically tree sap. And you see these once in a while being sold in different places. Some people even make jewelry out of these. Okay. One of the neat things with this is that the body is pretty much preserved there. So you can see things that you couldn't see in the other type of fossils, like the color of the animal, the internal structures. Okay. Third one, frozen. Okay. So this is not as common, and obviously only in certain parts of the world where it's cold and icy. So if something dies, like this woolly mammoth or mastodon, it's preserved, and the like actually it's a woolly mammoth, preserved, and it can stay there for years until it's found. You know, now these are not as old as the ones in rock, but this is probably something like 10,000 years, but it's still a fossil. Number 4, petrification. This is the same concept as the first one, where the minerals replace the tissue, the organs, the muscles, the skin. Okay, so as the minerals fill in the space in the tissues, like bone, so that like in case of these trees, the wood becomes rock. Then this is one that's not found all over the world, just found in one place. In LA, there is an area called the La Brea tar pits. I don't know if you've been there. It's a really cool place. It's right downtown LA. It's at least, I think, 38,000 years old. Basically, it's these tar pits. Okay, so these are statues, but this in the background is real. It looks like a pond, but there's tar, asphalt just coming up from the ground. Okay, and if you look at the diagram here, okay, you have the parking lot, the museum, and then you have these tar pits. Okay, has any of you gone there? No? If you do, I mean, if you have a chance, go. It's really, really cool. And you can actually walk to see where they are digging. Okay. Okay, someone has gone. Good. So it's basically an old trap. These pools had or have this tar, and animals that would walk over would get stuck like quicksand. And imagine that it was a prey, like a deer. It would get caught, screams, thrash, all that, and that would attract the predator. And then the predator would come and also get stuck. So you have prey, predator, prey, predator. You have all these animals that show you the history of what was going on in California, in that one area. They have a lot of these fossils of these wolves called dire wolves. It's these really big wolves that were really common. And you could see them going there because you have a lot of prey, and they would go to try to get it, and then they would get stuck. Okay. So that's the 5 ways that, or the 5 common ways that the fossils are formed. Now, one of the things that we can also learn from fossils is a lot of behavior, because you have animals or have fossils of animals that give us an insight into their biology, or the animals were doing something when they died. Okay, so this is showing you the tar pits. This skull is a dire wolf. Okay. So looking at the behavior ecology. Okay. For example, on the upper left, you have a footprint. This tells you about how the animal walked, what kind of, how the hips were formed. So a lot of information just from one simple footprint. Okay. Leaves tell us, what the climate was like, what plants were around at that time. Okay. And also trace fossils. like the footprint, or a shell, like on the lower right. Okay, it may have sediment in the shell itself. It just tells us an insight into the biology or behavior, or even the ecology of that animal or that environment. Okay. Something else that you probably seen in a museum, because they always end up in the museums, are animals in the act. Like these animals died while they were doing something. Okay. I took this picture, so there's what you see here is the reflection of the glass. I think this was either the LA Museum of Natural History or the New York. But what you see is, you can see the contents. Look inside, look between the ribs. You see that what's inside there. So you could tell that what they had eaten, and so you could think about what their diet was, what kind of environment they lived, if that other animal that they ate was there. So it gives you a lot of ideas, of what the environment was like. Okay.
Is stomach content like that too old to sample to see what it was?
Yeah, I mean, basically, it just tells you like they were carnivores or they were herbivores. You know, um, you couldn't. You might be able to. You might be able to tell what animal they were eating. If it's a bone, that you can identify it, but you couldn't. Something really old, you couldn't. You could probably even date it. You can say how old it was, but you couldn't tell. Like, I mean, if they're all fossils, but you can kind of tell what, just in general, these animals ate these little ones. In other fossils, something similar to this is they could tell. For example, there's an animal called Ichthyosaurus. It looks like a dolphin, but it's not. It's a reptile. But that animal gave birth to live animals, to live babies, because they found the skeleton of a baby in its belly.
Oh,
So
wow.
they know that it didn't lay eggs. They know it was a live birth.
Okay.
But some cases, like for example here, so you have 4 trace fossils. Okay, let me see if I find these correct. Here you have some fish eating another one. Here you have 2 on the upper left. You have 2 turtles having sex. Here on the bottom left, you have looks like a reptile eating something else. And the same on the bottom right. You see that it looks like a dinosaur head. Here's the mouth, and then this little thing inside it. Okay. So again, this gives you an insight onto their biology. Okay. Here's another one, I think. Yeah. Again, this is from a museum. They found the eggs and the skeletons next to the eggs. So we know that they laid eggs, but we also know that there was some type of maternal care, like sitting and incubating. For example, like birds, they sit on the eggs and incubate them. But like sea turtles or turtles, they lay the eggs. The mom lays the eggs, covers them, and leaves. There's no incubation, nothing. The heat of the soil incubates them. Okay, so if you find 2 animals, which were probably the parents, next to the eggs, then okay, maybe they formed the pair like some birds do, and they looked out for the eggs, which means that they probably also took care of the babies when they hatched. So you
I have a
learn.
question.
Yeah.
So I see this picture here, and it's always been like a question with obviously a lot of like you see this representation of these animals here. So yeah, you look at ones maybe protecting the eggs, the ones defending it. Like that's just the presentation that these people put on. It's not necessarily a representation of how these animals or these vertebrates behaved. It's just what they present to you, right?
Yeah, but it's based on how they were when they found them. So to be honest, like most of these fossils are actually copies of the originals, but how they found them told them like they were found near the eggs. So that's like they wouldn't do this if the two adults were not found near the eggs. They wouldn't have
But
done that.
but it's an assumption. It's not. I mean, like you can assume that they may be protecting the eggs, which is great, right? But there's no telltale sign that that was what they were doing, right?
Oh, right. I mean, they could have just been walking by the
This
nest.
picture right here that you're showing is this one's protecting the people that are
Hold on.
coming.
Can you mute the dog? Whoever,
These
can you
dogs.
guys mute him? No, I mean, yeah, it was probably, I mean, they could have been two animals walking and died by another nest that was there. Okay. But that would be kind of rare because no, they have more than just two fossils of this species. So they wouldn't just do that based on one sample fossil.
So they've got a history of something that they know the behavior of it, and to present this to us to show us how they act, right?
Right. And then if you were at the museum, you know how they have the nameplate with the species or at least the family, you can look it up and then they'll tell you the fossils. There's not one that you're looking at, but then there's another, like basically like imagine that you were writing a paper about that animal. You can look up all the fossils that exist of that animal and you get an idea. Yeah.
Oh, okay, thank you.
Sure. Let me see. Okay, now this is poop, fossil poop. So what this does is the diet also. So when you get poop, you can analyze it, pollen fossil, plant fossil, and you can tell what the animals were eating. Okay. Now some fossils are called index fossils. Okay. And that like these, for example, these are a type of trilobites. That's the genus Phacops. And we know that they were about 400 million years old. The time is the Silurian and the Devonian. Okay, so if you are digging up a fossil of a fish, and you find these next to it or in the same layers, you know that the rock you are digging is about 400 million years old. So just an example of using fossils to date other fossils. Okay. Now, one of the famous fossil beds, there are about 2 or 3, but this is probably the oldest as far as being the most famous. Okay, it's called the Burgess Shale. Okay, and this is up in Canada. A guy named Walcott that found it. Again, it's the Cambrian. Remember when I told you there was a Cambrian explosion where we go from no life to a lot of life? So he found this fossil bed just on the side of the road that had so many, I mean, something like 60,000 specimens, okay, in this fossil bed. And when you look at this, you see the different types of animals and plants that existed, and, again, all, in the ocean, but you see that the variety of life was really, let me just maximize this. The variety of life was really amazing, from porifera, there are sponges, arthropods, or the insect-like annelida worms, all these different worms, mollusca. And again, the Cambrian is a period where when you watch the movie, you guys still there? Hold on.
Yes, we
Sorry,
are.
sorry. My computer just flashed. So the Cambrian is a period where you went from having no life to having life. And when you watch those videos, you'll see that, wow, it was in the bottom of the sea. And I don't want to spoil the movie, but basically Earth was frozen. It was called Snowball Earth. And all the oceans were covered with ice, but under the ice, there was life. And then when it finally melted, all this life that had survived under the ice had evolved into all of this. Okay. There is a book called Wonderful Life by Stephen Jay Gould that talks about this fossil bed, the Burgess Shale. Okay. Just showing you some of the animals that existed. This one is funny. It's called hallucigenia because they didn't know if the animal was upside down or right side up. They didn't know which way it was, top or bottom. Okay. Here's another one, Wewaxia. Another one, Picaya. Now, Picaya is an early chordate. It has those 5 characteristics. Okay. Now, another fossil bed, that's a more recent one, is in China, in the Yunnan province. And this is a really good fossil bed for dinosaurs with feathers. Okay. For a long time, birds were considered a separate group. And then people realized, well, birds are related to reptiles. And, this is probably like 30 years ago. Some said yes, some said no. They were fighting back and forth. Well, now there's no doubt the birds are reptiles and they're actually dinosaurs. So they are descendants of dinosaurs. Okay. And again, this particular fossil bed is really rich in these weird looking just imagine like a dinosaur the size of a bird with feathers either on the wings, tails, kind of weird patterns. Some just have on the tips of the wings, some have on the heads. So you see the whole movement from no feathers to feathers in these dinosaurs. Okay. So the last thing I want to show you on this PowerPoint is how these fossils are made. Again, this is just so you have an idea. Imagine, again, there are new tools recently where you can kind of scan through the rock, but this is how the old way, and still nowadays how it's done. So you have this rock and you see the little thing here sticking out and you like, oh, that could be a fossil. Okay. So you get your tools, okay, and basically you're going to chip the rock away. And sometimes you have to do this through a microscope, like on this upper right. And if you go to the La Brea tar pits in LA, they have this thing. You can see the scientists or the paleontologists digging the fossil out of the rock, and you can see them using these type of microscopes. Okay, so basically you start chipping the rock and you can see the darker fossil, and you just keep going and keep going, keep going until you kind of have the whole animal there. And you see all the little scratches on the side where you think you might have something, but it's really not there. So then when you're done, then they clean it up, make it look nice. And again, usually they make a cast, and that's what they show to the public, or what they sometimes make copies for other museums. Okay, the originals are usually in the back somewhere. Okay. All right, so let me stop sharing. So now I am going to stop recording because it's been